It's a great pleasure to be here, to see you all, to be in such a distinguished company, and to be in Porto again. I think it's my third visit, and I've always enjoyed being here. Now, I've known Francesco for quite a long time, and he gave me quite a tough topic to talk on, but it's been great fun putting some ideas and some slides together. So I have a few disclosures, but none of them are particularly relevant to today. So this is the topic, structures and mechanisms. So structures and mechanisms controlling anesthesia-induced unconsciousness. So we all, in our training, learn that anesthesia has several components. And the main one, of course, is unconsciousness, or hypnosis, we often call it. But there's many other aspects of anesthesia that are important for the surgeon: immobility, so the patient doesn't help with the operation, and analgesia, we commonly call it. So we're going to focus on unconsciousness. So unconsciousness is obviously absence of consciousness, but what is consciousness? This is a really difficult subject to deal with. And there's a huge literature on it, and we can't go through it now, even though it's so interesting. But one of the early people to write about this and try and formalize some thoughts was, of course, Descartes. And Descartes believed in dualism. So he believed that the mind was separate from the body, mind and matter separation. And mind is a set of cognitive functions, or faculties they're called, of which one is consciousness. There are many others, judgment and so on, which don't necessarily need consciousness. But I think you'll agree that we've kind of moved away from dualism now, and we really believe that consciousness is a property in the brain. He believed that the mind was something above, he called it metaphysical, so it's not physical. The mind is contained in some other space that we don't really understand. But we really believe now that the consciousness and the mind is part of the brain. But the thing is, it's a very tough challenge to explain how molecules and neurons communicating with each other can generate the rich experience of consciousness. And there's this term, the hard problem of consciousness, which was coined by this man, Chalmers, some time ago, but he's still active. So things have moved on a little bit. There's a lot more information, a lot to help us understand consciousness, but in many ways this is where we are. You've got cells and they're communicating with each other, and poof, there's consciousness. But as I say, consciousness is a very rich experience. Here's a photograph from the early 1970s, a family picture with cousins and so on. And obviously, when I look at it, my experience is highly individual and different. And how do you know what I'm experiencing, and how do I know what you're experiencing, or if you're experiencing anything at all? These are very challenging concepts. But what's clear is that for me to experience this picture, there's a whole lot of different parts of the brain that are taking information from this and binding it, it's called, into an experience. So there's different colors, there's different shapes, different parts of the visual cortex are analyzing this and eventually saying, oh, there's faces there, and then accessing memory and saying, oh, those are my cousins, and oh, that little one is dead, unfortunately. And then there's emotion, and it's communicating with working memory, and so on and so forth. And this concept, among others, is summarized nicely in a recent article by some very eminent people in the field, starting with Melanie Bowley. And this was published in Journal of Neuroscience just last year. And it's a nice summary of current thinking in many ways, but also some challenging thoughts, showing you that we're still a long way to go, because the title here is, are the neural correlates of consciousness in the front or the back of the cerebral cortex? This is a very basic question. So we still have a long way to go. But what this graphic here is illustrating is really this binding issue, that there's different parts of this sensory cortex registering the signals, there's communication with the association cortex, and then there's eventually outputs to perhaps a motor area to move my mouth to tell you something about it, communication with the verbal parts in the forebrain to say what I'm going to say, etc. But we don't have time to carry on about consciousness, but let's just, for today, assume consciousness is a function of the brain. It arises from physical properties and processes in the brain. These are electrochemical, probably. It requires neurons that propagate signals, communication between neurons, summation and integration of signals, communication within systems, and then finally communications within networks and between networks. And these are some of the key sites that where anesthesia might be having its effect. Later on, you'll see we'll come up with these concepts of functional connectivity and binding. So I thought I would just kind of define them now. But functional connectivity, this is connectivity between brain regions that share functional properties. So we think in terms of networks quite often, and they can be quite distributed. So the default mode network, for example, which is when you're not doing anything much, it's got distributed components. And really what we mean by functional connectivity is it's a situation that we judge to be there when we see a temporal correlation in electrical activity, maybe with EEG, or within bold signal with functional imaging. And this is a correlation in the signal between spatially remote areas. And for there to be functional connectivity, we don't necessarily have to be able to say there's an anatomical connection between them. So there's no assumption of that. Now, binding is a different concept. It's what I referred to about that photograph. But binding is simply this idea that to be conscious, there needs to be communication, and there needs to be there's different aspects of the brain that are analyzing different sensory inputs, so different regions, and they're putting this together, and eventually there's some representation in the working memory. So these elements of neural information are bound together. This is a rough description of binding. All right. So anesthesia produces unconsciousness, drug-induced unconsciousness. And I remember when I began my training, people kind of thought that the brain, there's nothing going on. Well, that's not true. And during anesthesia, even quite deep anesthesia, the brain is not electrically silent. There's still electrical activity happening. It's just that this activity is unable to generate consciousness, maybe, or probably. But you can never be sure that someone is not conscious, because how do you know? They may not be able to say it or whatever, but it might still have been. So we think that anesthesia interferes with this electrochemical signaling, and we're going to go through some of the data of the various places where anesthetic drugs are interfering with these processes, and might be the places where anesthesia is generating unconsciousness. So the story has been growing over the last few decades, and it started when I began my training. It was all about the lipid membrane. Then not long after that, we started talking about receptors, then systems, communication between different parts of the brain, and finally connectivity. And now, interestingly, you'll see towards the end of the lecture, we seem to have gone almost back to the membrane again, with theories based on effects at the membrane. So when I began my training, we were told that the Meyer-Overton hypothesis explained how our anesthetic agents worked. They all worked through a very simple mechanism, we were told. They were lipid-soluble. The more lipid-soluble our drugs were, the more potent they were, so a lower MAC value. And this was because they diffused into the hydrophilic parts of the membrane, and they changed the properties of the cell membrane of neuronal cells, and this interfered with the function. So it was a very simple, almost physical explanation of how our anesthetic drugs work. There was some evidence for parts of it. There was the pressure reversal theory and so on and so forth. But that's what we believed. And even there's still people that believe in this, and who knows, maybe it's partly true. And in 1998, Al-Kaya published one of several studies that he did using one or other type of functional imaging. This is one where he looked at glucose metabolism across the brain with different depths of anesthesia, with either propofol or isoflurane. And the color on these slices of the brain is proportional to the amount of activity, cerebral glucose metabolism uptake. And essentially what you're seeing from the top, the awake state, down through deeper and deeper anesthesia, eventually when the BIS is 34, there's global suppression. Many people took this almost as confirmation of the simple theory of the Meyer-Overton explanation, because in the bottom picture it seems quite universal and symmetrical or across the brain. But actually when you do a subtraction analysis and look at the differences per area or per pixel between awake and asleep, then you'll see there are quite big differences across the brain. Now, we've moved on a lot, and for a lot of the time since the 90s, we've been mostly focused on effects of our anesthetic drugs on receptors. So these are protein elements and voltage and ligand-gated receptors. And one of the pioneers of this work was Nick Franks. He showed in the 1980s that anesthetic drugs have dose-related effects on proteins. He initially studied firefly luciferase, but later did work with all sorts of other receptors and showed very nicely that the GABA-A receptors, so that our drugs are mostly having effects there as agonists, so they potentiate these receptors. And there was a very nice review that he wrote in 1997 in Nature on this hypothesis. Since then, there's been a lot of work, some of it really interesting, or all of it interesting, and some of it some really fascinating techniques. But it's been drilled down in much more detail. And some of the big advances have come from genetic manipulation studies. And here, they've often used knockout or knock-in mice, where they can change the structure of one of the components of the GABA receptor and see what effects this have. And generally, they'll do these genetic manipulation, but the mice are otherwise fit and well. And with these kind of techniques, people like this group here, they've been able to show that different components of anesthetic action are probably mediated by different isoforms of the subunits of the receptor. And these involved these knock-in and knock-out mice, and they would give them doses of anesthetic and look how long it took for them to get their balance and the ability, or how long it might take them, for example, to lose the ability to walk on this rotor rod thing. So for those of you interested in this, is very interesting studies. Now, over the years, they've been shown that our anesthetic drugs don't only work on the GABA receptors. In fact, they have widespread effects. And on the neurons, there are lots of receptors, of course, within the whole brain. And a lot of our drugs have effects at very many places. And here's a summary that Al-Kai published in a nice review he wrote in Science in 2008. But what you'll see is that none of our agents have pure effects. Although generally, when we look at them, they're either potentiating inhibition, so they'll be GABA-A agonists, or work on glycine receptors, or they're inhibiting excitatory pathways, so on the NMDA receptor, for example. But it's very difficult to know, with all these effects, which ones are key for anesthesia, and we've still got a long way to go. It's a very interesting work in 2006 that Ira Rampal published, another real neuro-anesthesiologist and one of our heroes, really. He showed that in one kind of cell, from one part of the brain, from the amygdala of a rat, isoflurane had effects on 269 proteins. So our drugs obviously have very, very many effects. They reduce the metabolism in the muscles. They change what's happening in the kidney and the liver. But obviously these effects aren't important for anesthesia. So that's one of the big challenges, is to know which are the ones that are interfering with consciousness, which ones are relevant. Interestingly, in this paper, Rampel, they did a nice cartoon showing a summary of all the different genes and proteins that they saw changes in that have effects on synaptic transmission. And this is the figure from their paper. And this is was almost prophetic, because there's been recent work showing that maybe our drugs are really having important effects, presynaptic effects on exocytosis. But that's moving ahead now. So we've started from membranes, we've gone to proteins, but along the way, people have also been looking at whole neuronal pathways. Here's something from the 1990s, and this work by Angel. They had implanted electrodes, and they gave these animals anesthesia. I think it was urethane, and it was urethane, and then they were able to show changes in electrical signaling and potentials along the pathway from the cuneate nucleus, the thalamus, and the cerebral cortex. And he came up with this concept that there's a kind of a switch at the hypothalamus, at the thalamus, I beg your pardon, and that signals were coming, but they weren't being transmitted further on from the thalamus during anesthesia. And this eventually crystallized into the thalamic switch kind of hypothesis. And Al-Kaya, he's one of the strong believers. I don't know if he still is, but he was a big proponent of this idea. And he did this very nice summary of a lot of the data that was existing at the time in this, which was a chapter in a book in 2005. But essentially, he looked at the results of different modalities of functional imaging with different drugs. So each slice of the brain shown here is from one study. So for example, in the top there, it was looking at sevoflurane, where regional cerebral blood flow was measured or quantified, and that would have been then with positron emission tomography, probably with labeled oxygen. And then each of these graphics is showing you the difference between conscious and unconscious, and where the biggest changes in the flow or metabolism have happened. And so as you go around, you see all the different drugs, and whether it was glucose or metabolism or cerebral blood flow, there they are. Is dexmedetomidine, clonidine, propofol, etc. What you immediately see is that the pattern of changes is different for the different drugs, and that's very interesting. But Al-Kaya did a conjunction analysis. So he looked which area is affected by all drugs with all techniques. And he showed that in all of them there was an effect at the thalamus. So the thalamus does seem to be an important site where there's anyway at least a uniform and consistent change. To me, it's not clear whether is that where the switch is, where the big consciousness man is switching it on or off, or is this a downstream effect that you just see the most consistent effect there when you lose consciousness? This is not at all clear. Now we're talking about pathways and work that's been done. Here's other work looking at different drugs and sleep pathways. And there seems to be good evidence. This one was published in Nature Neuroscience by, I think it was Mervyn Mays' group, but they used micro-injections and electrodes in these animals, and they were able to show that propofol and some other anesthetic agents seem to have effects on the natural sleep pathways. And in red there, in the middle, you see an important sleep pathway. It starts with the locus cerealis in the arousal, ascending arousal system, goes on to Vilpo, and eventually the tubomammillary nucleus. And GABA effects at these sites are essentially activating a sleep-promoting pathway, and that results in reduced release of histamine at the cortex and sleep. So here it is. And it was a very interesting study. They also not only did injections of drugs like propofol and thiopentone, they also used muscimol, which is another GABA agonist. It comes from mushrooms, and it causes hallucinations and so on. But they also gave these animals sometimes gabazine or not, and gabazine is a GABA antagonist. And essentially, they were able to show that for some drugs, these effects were reversed by gabazine and some not. But clearly, this is just one aspect of how these drugs work. Because, for example, muscimol, although it can cause sedation and hypnosis, it also causes hallucination and other phenomena. It's abused sometimes, this drug. So obviously, it's having other effects, because usually hallucinations involve some kind of activation. We'll move on. The same group also did work with dexmedetomidine, and were again able to show that dexmedetomidine seems to have effects on this sleep pathway. And maybe dexmedetomidine's effects are more pure, because clinically, dexmedetomidine sedation certainly seems to look to our eyes much more like sleep than, say, propofol. In this pathway, this graphic from their article, sadly it didn't print very well in the journal, but on the one side you can see the perpendicular nucleus, and this is an orexinergic nucleus. And the picture is complex and is going to stay complex and maybe even get more complex as we go with time. But what this group did, again looking at pathways but in different ways, they were able to show that that is also an important part of the pathway, and they were able to show that this orexinergic area has a very important role in emergence from anesthesia, but not in induction. So again, this is suggesting that the anesthesia is a much more complex, we know this, but much more complex, say, than physical effects, because they seem to be path independent. So the one process is not simply the mirror image of the other. And what they did was, in this experiment, they used mice and flies, and they were able to measure brain concentrations of drugs, and they were able to determine whether the brain concentration was the same at loss of consciousness and return of consciousness, or at least 50% probability of loss of consciousness and return of consciousness. And what was very interesting was that not only did they show that this brain concentration was different during induction and emergence, there was a neuronal inertia, so the concentrations had to fall much lower before the animals regained consciousness. But that difference was different for different anesthetics. So for propofol, there have been various studies, one from Groningen, and they have some conflicting evidence. But just looking at the data, it seems to me that the fat solubility of the agents might influence how much inertia there is. And this is then maybe suggesting, if you're talking about fat solubility, maybe that is a membrane effect. But we don't know. So we move on from pathways now. There's been a lot of interesting work along the way, simply looking at regions of the brain, regions where we know that have specific cognitive effects. A lot of the time this is involved either functional imaging with positron emission tomography scanning, or with MRI, with functional imaging, with functional MRI. And in this interesting study here, they administered propofol until loss of consciousness. So they gave a slightly slow infusion rate, and the people gradually went off to sleep. The volunteers, I assume they were residents, but they were volunteers. They sort of volunteers. But they got verbal stimuli, and they had these functional imaging. And so what you see on the top, you might not see it, I suppose, is the network of areas activated by the verbal stimuli when the people were awake. And it's the typical areas you would expect: the primary auditory cortex, some association areas, and an inferior frontal. And then you see, as they were becoming sleepy but still responding, it's shrinking down. The primary auditory cortex is still being activated, but the others less. And then after loss of behavioral response, so when they heard a stimulus, they were supposed to click a button. So after they stopped clicking the button, very interestingly, the primary auditory areas, Herschel's gyrus and so on, are still being activated, but they were unconscious. And this is concordant with what I said earlier. The brain is active, it's just that the information isn't spreading and leading to behavioral changes. Only when anesthesia was very deep were there no more activations. So a lot of the work that's been done to try to understand anesthetic agents and how they work has involved functional imaging. And here we go back to Al-Kaya. He was looking at very low doses of sevoflurane and the effects that this had on certain kinds of emotional memory. And you can see he used there in the top left graphic, basically they looked at baseline and then low concentrations of sevoflurane. The maximum was 0.25%, so 0.25%. And so what he was doing here mostly was looking at whether memory was functioning or not, and then looking at communication between different areas of the brain, here mostly the limbic system. And he was able to show that sevoflurane had very specific effects on the communication between elements of this limbic system. And some areas seem to, the effect seemed to be stronger and some areas less. So the communication, sometimes communication is correlated and sometimes anti-correlated, but on the graphic on the right, it's showing that there was a big change in the communication, especially between the amygdala and the hippocampus. So there's a big argument in the literature about whether anesthetic effects are affecting top-down processes or bottom-up processes. So top-down would be correlation, communication between higher order areas like association areas and so on, versus primary auditory or sensory areas. And most of the evidence at the moment seems to show that the effect when anesthesia induces unconsciousness, this is associated with reduced top-down communication. So this is sometimes difficult to understand because we would imagine that it's the other way around. But on the other hand, we do know that after loss of consciousness, when anesthesia is still shallow, there is still activation of your sensory areas. But we would, I would imagine that maybe there's still communication this way, but not the other way. But the evidence is against this. Here's one study where they did show this, and that they thought that it was mostly bottom up, but a lot of the evidence is the other way around. And here's some work published a few years ago, again by Melanie Bowley and some very eminent researchers, and they looked at connectivity changes during propofol-induced loss of consciousness. They examined different models, and this was a study where they looked at EEG changes. So in this model, which was the one that fitted their data the best, they're looking at communication between thalamus in the bottom, parietal and frontal areas shown by P and F. And on the right, they're showing arbitrary units, but excitability of thalamus and connectivity frontal to parietal. So what they were able to show was that during light sedation, they thought that the activity in the thalamus was slightly increased. I'm not sure quite how to explain that, and that this went even further after loss of consciousness. But when they looked at connectivity, they showed that the front to back communication or connectivity was lost. That seemed to them to be the signature of loss of consciousness. So this is then more behind the central sulcus, as you know, is mostly perception and sensory areas, and so it's a communication for more association and executive functions. That way back was interrupted. And this has been shown in many different ways and by different groups. One of the big stars in this field is George Mashore, and he works with various groups. He's a person to watch, if you ask me. He's published a lot of work. He recently had a nice editorial in Science, and I think he's a dean of research in a very big university. So it's nice to see an anesthesiologist in such an eminent and prominent role. But this work was people from Korea. They looked at so front to back and back to front connectivity in the EEG with 3 different drugs, with ketamine, propofol, and sevoflurane. And they did a connectivity analysis, but they started with a traditional EEG analysis, dividing the EEG into different frequency bands. And in each of these little graphics, it's time on the x-axis, and on the y-axis, the power in that band. And then the gray area here is when they got anesthesia, and at the end of it, they lost consciousness. Thank you. That's okay. Don't worry. Don't worry. Yeah. Okay. So if you do a traditional just EEG frequency analysis and divide it into bands, what the first interesting thing is that during loss of consciousness, for each of the drugs, ketamine, propofol, and sevoflurane, there are very different things happening in the power in these frequency bands. So for example, in the gamma band, so higher frequencies, 40 plus, with ketamine, during loss of consciousness, the power is increasing. With propofol, it's decreasing, and with sevoflurane also. Okay, so we know the ketamine works at NMDA receptors, it's working at different places perhaps, but loss of consciousness is associated with very different changes in these signatures. But when they looked at front to back, so feedback and feed forward communication, they saw quite a consistent pattern. So here are the 3 drugs. The gray or blue, I'm colorblind, is the period when the anesthetic was given. And so feed forward is in blue and feedback is in red. And what you can see is that this was very consistent. So during loss of consciousness, for all these 3 drugs, which work in different ways, sevoflurane has more effects on potassium channels and other channels than propofol and so on. But for all of them, there was a marked change in a feedback, so from front to back communication in the brain. And this was consistent across the 3. And this has been reproduced in different ways. This group here, again with George, but with different researchers, again looked at in the frequency bands, and they're looking at long-range temporal correlations across the brain, and they were able to show that loss of consciousness was associated with quite specific changes. This is looking at correlations again, front to back, in activity. And again, they showed that with unconsciousness, there's a decrease in occipito-temporal correlation. So there's a whole interesting aspect of science called information integration theory, and this ties in nicely with that, but we don't have time to get into the details of that. So I see that my time is nearly up, but what's interesting to me is that although for a long time we've been saying that membrane effects were probably not relevant to anesthetic effects, things seem to have gone back a few steps. and we seem to be, in some of the work is going to, again, more simple explanations. Here is one. This is a nice review of some of the evidence published just 3 years ago, looking at dendritic signaling. So this is through in pyramidal neurons that go through different layers of the cortex, and showing that there's widespread effects of different anesthetic agents along these pathways, and this is a possible explanation, is offered in this article anyway, on how this might be how front to back or feedback inhibition is occurring. But more interestingly, and back to the membrane again, there's been some work showing that maybe the anesthetic agents are, some of their effects anyway, are really mediated by effects in the lipid bilayer. A lot of it revolves around exocytosis and effects on the mechanism by which neurotransmitters are released into the synaptic cleft. So this involves exocytosis. It involves vesicles that come down, little packets of your neurotransmitter, and then there's a whole mechanism, the snare mechanism, different proteins that pull it down, attach it to the membrane, and eventually open it for this package. And this molecule here, syntaxin, it seems that propofol seems to, and some other drugs maybe, seem to have quite specific effects on this, but that it's mediated by effects from the propofol while it's in the lipid bilayer. So we, in some ways, we seem to have come back or gone full circle. But here's one of their recent papers showing, they call it trapping of syntaxin 1, so that molecule can't move around and be involved in bringing these vesicles. So they're saying that it's reorganizing these syntaxin molecules, making them less mobile, and that propofol maybe is impairing the presynaptic release of a whole range of neurotransmitters. So back to a more non-specific effect rather than specific effects. Now, I've done some really interesting work where they give different analogs of propofol that aren't anesthetically active and show increased mobility in these syntaxins, and a range of interesting work with this technique. And for those of you really interested, they've developed a technique of showing single molecule imaging. So they attach a phosphorescent molecule onto syntaxins, and with microscopy, they can show the movement of single molecules. Very interesting work. So this is just a nice summary from this paper showing that these syntaxins on the left with propofol become immobile, and that this impairs exocytosis and release of their molecules. And so they've come up with a very interesting theory. And it might explain many of the things that happen during anesthesia. I'll get back to that in a second. But essentially what they're proposing is the following. They're saying that there's a sleep pathway where our drugs are working on GABA agonists, and maybe dexmedetomidine and its effects as an alpha-2 agonist and so on. And this is a kind of sleep, sedation, wakefulness part. And that this is important, and maybe the orexins, during induction and emergence of anesthesia. So they're saying maybe those are specific effects and those are important, but maybe there's another important effect, and that for anesthesia, during surgical levels of anesthesia anyway, our drugs, propofol and others, are having, this is their hypothesis anyway, effects somewhat non-specific in the lipid bilayer, so almost back to the Meyer-Overton hypothesis. But these are the effects then that maybe bring the other components of anesthesia, like immobility, analgesia, loss of behavioral responses, loss of memory, and so on. It's worth looking this paper up, because they also give rationales for how this hypothesis might explain why there's this neuronal inertia, why drug concentrations have to be much higher in the brain for loss of consciousness and why they have to drop very low before you get return of consciousness. And I think, I've not seen it written anywhere, but I wonder if there's something here that might explain how awareness happens. That maybe someone, because when you look at the awareness study, some of the patients weren't paralyzed. In the Sundin study, 4 of the patients who weren't paralyzed didn't move, but still had a verifiable account of what happened to them during anesthesia. So you kind of wonder whether maybe the one pathway was inhibited still and the other not, and so they returned to consciousness, or at least memory functions were working, but they lay still. It's this wild hypothesis. So we've kind of finished on maybe a mix, but maybe back to the lipid bilayers. But before finishing, I'd like to return to my favorite theory from this man who also looks like a wizard, and he works with Chalmers, who also looks like a wizard, and who coined that term, the hard problem of consciousness. But this is a beautiful theory that he used to explain anesthesia and unconsciousness, Stuart Hamerhoff, and this involves quantum interaction, so it involves quantum physics. And this is an explanation for how there might be communication between remote parts of the brain, not with signals going along nerves, but quantum interactions between molecules or structures in the front and the back. But that's not a theory that seems to be talked about much. So on that note, I say thank you for your attention. I think there's a few minutes for questions.